Okay. So just we have discussed about the binomial nomenclature and how we can use this particular binomial names to describe the plants. And these names how they are internationally accepted. Now the next topic under the international code for botanical nomenclature we are going to see is the rules of the ICBN. Okay, that are what are the rules of the ICBN are there in the by various scientists over a period of time. Now these are rules have got perfected over a period of time, and we should follow these rules as well while giving the name to the plant. Okay, now what is the difference between the binomial nomenclature and rules of the ICBN? Okay, these are rules of the ICBNs are very broad rules. Okay, and they were giving the minute details for each and everything. That is how to name the plant, how to give the author citation, how not to use the repetitive names, right? So all these rules of the ICBNs are there. We should understand them very carefully because UPSC can ask you alone what are the rules of the ICBN, or they can alone go for the topic that is the binomial nomenclature. Okay, so now consider the rules of the ICBN. See, we are dealing with the paper 1 and the chapter number 3, that is the phenerogams, under which we have to study gymnosperms as well as angiosperms, and we are at here International Code of Botanical Nomenclature, right? Now consider the introductory slide. See, the rules of the ICBN. Under the rules of the ICBN, the first rule we have is the rule of priority. Okay, so as the heading suggests, the priority. Okay, as you know, we use this term often that to which thing we have to give the priority and to which thing we don't have to give the priority. Okay, so the same meaning is also here. The rule of the priority is there during the botanical nomenclature. If there are the plants, one same plant, and this particular one same plant has the many names, okay, then what should be the priority, which name should have the first priority? Regarding that, we have the rule of priority under the ICBN. See, it has been found that the number of published names of the various plants exceed the actual names of the taxa. Okay, so there is a single taxa, and this single taxa, for example, A taxa is there, and this particular taxa has so many names published in the literature. For example, in the India, you find one name for one particular A plant, and this particular A plant is known by a particular name in the India. But in the Japan, this particular same plant species is described by the same other name. The same is true for the Germany. The German is publishing the plant material, but it is giving the very other name in the German language to this particular A plant. So what happens over a period of time when there was not a regulation regarding this particular naming system, we have the so many names for the single taxa. Okay, so the names are many and they were actually exceeding the taxa. Okay, so whatever the discovered taxa today we find, these taxa have so many names that number of names are quite higher than the number of the taxa. See, this is because the several names have been frequently attributed to the same taxon. Okay, that is the same A species is having the many different names throughout the world. Okay, throughout the world they were using the different names to describe this particular plant, one single plant. So same taxon is having the several names. Hence, it is very essential to have a way to decide which of the several names should be selected for a particular taxon. So for example, this particular A taxon has the 4 names, okay, published in the research. Okay, so first name is coming from the India, second name is coming from the Germany, third name is coming from the Japan, and the fourth name is coming from the America. Now the question arises, which one is the legal name? Okay, and what are the rules regarding to decide which one is the legal name in this particular scenario? For example, the species A is described by the all 4 countries, and all 4 countries researcher has given their own name. So which one to give the priority? What are the rules regarding that? So the ICBN provides the rule of priority in such a condition that there should be the priority of one of the 4 names, right? And who should decide this priority? The rules of the ICBN should decide this priority. See how? See, this is where the principle of priority comes into the play, which insists that each family or taxon of the lower rank with particularly circumscription, position and rank can bear only the one correct name, and each taxon is to be known by the earliest name validly published in the taxon. See, very, very important content here. See, what ICBN says about this, in the principle of priority, they said that the A plant should have only the one correct name and not the 4 name. Okay, it should not have multi names, it should have only the one correct name, and that correct name should be the earliest published name. Okay, in this example we have told you that India published first, then the Japan, then the Germany, okay, then the America. Then in such a case, whoever first published this particular plant, for example, you consider the year, in which year this particular publication is there in the India, Japan, Germany and America. So the earliest publication, if there is earliest publication, one of the 4 should be the earliest publication. The earliest publication should be considered as a valid name. Okay, so what is the principle of priority here? In the principle of priority, when the single species got the so many names throughout the world, we will find that which name is published earlier, or which one is the earliest name given to this particular A species. If you find the this particular A species has given the earliest name in the India, that name should be the valid name. See, for example, here we have the Polygala L, which is published in the 1753. Then we have the Polygala Neck, which is published in the 1768. Then we have the polygloids Augusti, which is published in the 1770. Okay, all these last names, L here, Neck here, Augusti, are the scientist name, and these are the genus name. Okay, so Polygala, this was first described in the 1753, and Polygala L, this name is given. Then the same species describe Polygala Neck in 1768, and then same species Polygloides Augusti described in 1770. Then according to the principle of priority, the earliest name, which is called the Polygala L, is the correct name of the genus, because it is published in the 1953 and it is earliest one. Okay, and then the latter 1768 and 1770 came there. So the earliest name, whatever the earliest name is there which is published, that name can be considered as a valid name. Okay, and this is the principle of priority. For example, Polygala L. Okay, for example, Polygala L is considered as a name, a valid name of this species, because it was published earliest. Okay, and later published name should be disregarded. Okay, so this is how the principle of priority can be used. In the principle of priority, if the single one species has given the so many names, then in such a condition we should use the name which has given in the earliest time, that is in this case in the 1753. Okay, see, the second one is the rank of the taxa. Okay, so the second rule of the ICBN is the ranks of the taxa. See, the taxon is referred to as the taxonomic group of any rank. Okay, we have discussed what is the taxon already. See, the taxon can be placed at the species, genus, family, order, class, phylum, kingdom level, and all these ranks can be considered as a taxon. Okay, so taxon can be considered any one of the following these, and these species, genus, family, order, class, phylum, or the kingdom can be considered as a taxon. Okay, see, the every individual plant is treated as belonging to the number of successively higher ranks with the species as its basic unit. Okay, so every species which is described, every plant species which is described, it should have the lowest species name, and it should belong to the successively to the genus, family, order, class, phylum, kingdom, etc. Okay, so it should be placed systematically as we go higher in the classification, that the species, at the species level it's a basic unit, and after the species we have the genus, then the family, then the order, then the class, then the phylum, and then the kingdom. So the every individual plant is treated as belonging to the number of successively higher ranks. Okay, because every species actually belongs to the several higher rank, because it should have the species name, it should belong to the some of the any genus, it should belong to the family, it should belong to the order, it should belong to the class, it should belong to the phylum, and it should belong to the kingdom. Okay, so in this way, throughout the hierarchy, this a particular plant species has its own belongings. Okay, because if it is a species, it should be the part of genus, because generic name should be there, species name should be there. If the species name is there, it's a species, it should have the generic name, that is it's a genus. And this species, this particular species which has a binomial name, it must have belong to the sum of the family. This family should belongs to the order, right, and to the successively progressive hierarchies, right. So here the rank of the taxa should be accurately given, and this taxa should possess the higher ranks as well, and should have the basic classification at the species level as well, right. See, the principle of ranks of the taxa in the ascending sequence is the species, genus, family, order, class, division, and the kingdom. We have discussed this thoroughly in the last lecture. The ascending sequence for this particular taxa, how to follow it? It should be from species to genus, because it is ascending, right? Genus to the family, family to the order, order to the class, and class to the division, and division to the kingdom. So in this way, we should give the ascending sequence for the any find out new species by giving it a ranks in the taxa. Okay, so what is the rule of the rank of the taxa? That the rank of the taxa should be given in the ascending order. First species, then genus, then family, then order, then class, then division, then kingdom. Okay, and this sequence should be strictly followed. Okay, and this sequence should not be changed, because these are the rules of the taxa, ranks of the taxa, and according to this sequence only you should give the ranks for the taxon. Okay, if the greater number of the ranks of the taxa are required, for example the species is exceptional species and you require the more ranks. Okay, for example here the species, genus, family, order, class, phylum, and the kingdom, these ranks are not sufficient and we have to add more and more ranks. Then in such a case, what you do, these are made either by adding the prefix, sub to the terms denoting the ranks. That is, we should use the sub and we should give the rank here. For example, subclass can be there, right? Sub kingdom can be there, right? And even the sub species can be there. Okay, so if you find the exceptional species and you require some extra taxa, then you can add the subspecies, you can add the subclass, and you can add the sub kingdom. But order should be the same. Okay, order should be the same like this, right, from species to the kingdom. If you add the sub kingdom, it should become above the kingdom, right? If you add the subclass, it should come above the subclass, right? So while giving ranks to the taxa, it should give in ascending order. And what is the order? Species, genus, family, order, class, phylum, and the kingdom. This is the order. And if you require some extra ranks, you can add the sub prefix, okay, and give the extra rank as well, right? See, the third one is the typification. See, the code has greatly emphasized on the typification of the taxa in order to bring stabilization of the names. Okay, see, typification, this particular rule of the ICBN, this particular typification is given in the code for what purpose? For the stabilization of the names. Okay, the name should be the stable, it should not change. Okay, and for the stabilization of this name, the various kinds of the typification is given. Okay, now let us consider them. See, the naming of the taxonomic group is determined by means of the nomenclatural types, where the nomenclatural type is the element to which the name of the taxon is permanently attached. Okay, so generally we use the nomenclatural type. This nomenclatural type is an element, and generally to this element, this taxon, the name of this taxon is particularly permanently attached, either as a correct name, okay, or as the synonym. Okay, so firstly, whenever you talk about the taxon, if you find a new species, this particular new species is named by the nomenclatural types. Okay, and this nomenclatural type is using the element, any element, okay, which is present in the plant, and by using this element, this particular element is permanently attached to this particular plant species, right? Either there will be the correct name or it can be the synonym, that is the similar name. Okay, so whatever the correct name is there or whatever the synonym is there, this particular synonym is created due to the element of the nomenclatural type, and this element is particularly attached to that plant species, right? It is not necessary that the most typical or the representative element of the taxon. Okay, so maximum times we do not find any typical or most typical or the representative element, because majority of the time plant species do not show any speciality to attach a particular element to it. Okay, then in such a case we use the typification. See how we can use the typification. The very first is the holotype. See, it is the one specimen or the other element used by the author designated by him as a nomenclatural type. Okay, so whatever the author who is describing the particular species, he is using the holotype. Okay, so he has bring about the specimen, he has taken the specimen of the plant and he has describing it, and he is designated by him as a nomenclatural type. Okay, so that is the by using this particular plant material, its characteristic, we are going to name this particular plant, and this is the basic nomenclatural type. Okay, the holotype is chosen by the original author from the single gathering made by collector at the time. Okay, for example, he go into the field and he has collected the plant material. Okay, and by studying this plant material, the original name is given by the author. Okay, holotype, what is the holotype? Holotype is the chosen by the original author. Okay, that is the first author who have described that plant, and when the first author or the original author is describing that single plant which is gathered by him during his collection, he called it as a holotype. Okay, we should call it as a holotype. That is the first time the species is collected by the original author, and he has described this particular species, and by describing, he has giving this particular specimen as a holotype, right? Because why it is holotype? Because it was first collected by the original author. That is the first time species is collected by an original author and he described it. Then in such a case, we call that specimen as a holotype. Okay, see, it is very easy to understand what is holotype. Holotype is nothing but the original plant material collected by the original author. Okay, and if first time describe it, then that such a material is called as the holotype, and it is expressed definitely at the time of the original publication. So at the original publication, whatever the plant material is used, that plant material is called as the holotype, and by using this particular holotype, the nomenclature has given. Okay, so what is holotype? Holotype is the original material is collected at that time, and the nomenclature is done by using this particular original material which is called holotype, and the publication has occurred. Okay, so whatever the plant material which is originally collected and the publication is made or depending on this particular material, this original material is considered as a holotype. Okay, see the second one. the isotype. Okay, then what is the isotype? If the original plant material is considered as a holotype, then what is the isotype? See, isotype is any duplicate or the part of the single gathering made by the collector at the time of the holotype. For example, he is collecting the holotype, and during the collection of the holotype, any part of that single gathering is there, or the duplicate part of that single gathering is there, that duplicate part is called as the isotype. Okay, for example, the original material, okay, whatever the original material is collected, this original material, for example, we have collected this original material is considered as a holotype, and along with this material, the other parts of the material or the duplicate plant material, for example, this is collected, then in such a case these two materials is considered as a isotype. Okay, so what is the difference between holotype and isotype? Holotype is the original plant material through which the nomenclature and publication has done, and alongside with this first plant material collection, whatever the other material is got collected which are very duplicate or the similar or the part of it, then we call that material as a isotype. Okay, say, if see, for example, if a new species is based on the single gathering that consists of the four specimen. Okay, here to understand isotype and the difference between isotype and holotype, we have the example that, for example, if you go for the search of the new species and you have collected the new species, for example, you have collected this plant as a new species, and during this single gathering you have collected the four specimen. How many specimen you have collected? You have collected the four specimen of the same plant with the same field number placed on the four separate herbarium sheet. Okay, so four specimens were collected: the first specimen, then the second specimen, then the third specimen, and the fourth specimen. All these four specimen, that is 1, 2, 3, 4, are placed on the herbarium sheets. Okay, and the common number. Okay, for the all these four specimen, we have the common field number. Okay, so common field number is there, and this common field number is placed on this particular four sheets. Okay, whatever the four herbarium sheets were created, all these four herbarium sheets have the common field number. Okay, see, the original author will designate one of them as a holotype. Okay, so whoever the original collector of this plant material is there, or who is the original author of this plant material, he will decide the one of it as the holotype. For example, he has given the first specimen as a holotype, while the remaining three, that is the 2, 3, and 4, are considered as a isotype. So whatever the other three plant material which got collected in the single gathering during that process, the other three material are considered as a isotype, while the first material is considered as a holotype. Okay, so the first one is the decided by the author as a holotype, while rest of the three are the isotype. Okay, so this is the basic difference between the holotype and the isotype. See, the third one is the paratype. See, a paratype is the specimen cited with the original description other than the holotype and the isotype. See, what is paratype? Okay, and how it is different from the holotype and the isotype. See, paratype is the specimen cited with the original description. Okay, so whatever the specimens we are cited along with the original description other than the holotype and the isotype, that specimen is called as the paratype. So if we have some different kind of plant material at the time of single gathering or the collection, apart from the holotype and isotype, whatever the material is there, this material will come under the paratype. See how, for example, if a new taxon is collected in one season without flowers and fruit. Okay, so we have find out that there were the new taxon, and we have to collect it, we have to create the herbarium sheet of it, and we have to nomenclature, we have to give the name to it. Okay, then in such a condition, if the new taxon is collected in a one season when there were no flowers and no fruits on the plant. Okay, that is, we have taken the plant, but at that season this particular plant do not have any flower or the fruit. Okay, and the same taxon, the similar plant, you have collected in the different season with the flowers and fruit. Next time when you have collected this plant has flowers as well as fruits. So the same taxa, okay, the taxa is same, the plant is same. Okay, same taxa is collected two times. The first time when it was collected, it do not have the flower and the fruits. The second time when it was collected in the different season, it has the flowers and fruits. Okay, then the two collection will be given the two different field numbers. Okay, so this one have the one field number. Okay, field number is different for this one particular specimen, and this field number is also different because these are collected at a different times. So field numbers are different although they are belonging to the same taxa. Okay, first time when it is collected without flowers and fruit, and second time when it is was collected, it was given the it has the fruits and flowers. So the both of these plants have the different field numbers. Okay, so whatever the different field numbers are there, out of these two collections, the author will select one collection as the holotype and the other as and the isotype. Okay, so one collection is considered as a holotype and the isotype, while the next gathering, okay, whatever the second gathering is there, if the as we have discussed that there were the first gathering and the second gathering, during first gathering there were no fruits, no flowers, and during the second gathering there were the fruits and the flowers. So during this particular one gathering, if there were the two specimens were actually collected, the first specimen can be designated as a holotype and second one is the isotype. While this second collection, see, while the next gathering cited in the protologs will form the paratype. So this second gathering material along with the fruits and the flower, it is considered as a paratype. Okay, so what is the difference between the holotype and the isotype with that of the paratype? In the to create the paratype, the two times collection should be there, and while the isotype and the holotype can be created in the single gathering. This is the basic difference. Okay, so the paratype can be considered as a second gathering along with the fruits and flower. Then the fourth one is the scene type. Now, what is scene type? See, scene type is one of the two or more specimens cited by the authors of the species with when no holotype was designated. Okay, or it is any one of the two or more specimen originally designated as a types. Okay, so if we have no holotype, okay, no holotype is described, then in such a condition the plant material is considered as a scene type. Okay, so scene type is easy to understand. When a holotype is not designated, whatever the specimens are there, these are specimens are considered as a scene type. Okay, see the next one, that is the lectotype. It is the specimen or the other element selected from the original material to serve as a nomenclatural type. Okay, when no holotype is designated at the time of publication or as long as it is missing. Okay, so holotype again is not there, and if the holotype is not there and some kind of plant material out of this original material is used for the to create the nomenclatural type or to serve as a nomenclatural type, then that plant material is called as the lectotype. Okay, so you have bring about the plant material, the author has bring the plant material, but he does not designate holotype. Okay, that is the, he does not say which one is the original material, and whatever the material out of it which is used for the nomenclatural type or serve as a nomenclatural type, these are called as the lectotype. Okay, actually for this lectotype to occur, the holotype should be missing. Okay, and if the holotype is missing and we are using some part of the original material to name the plant, then in such a case we use the term lectotype. Okay, see, if the holotype is lost or destroyed, okay, that is the earlier there were the holotype, but it is for some reason getting lost or destroyed, the lectotype is selected from the isotypes. Okay, so whatever the other plant material is there, from this other plant material the lectotype is got selected. Or when two or more specimen have been designated as a type of the author of a species, the lectotype must be chosen among these types. Okay, so if they were showing the two or more specimens are there, all were forming the isotopes. Out of these isotopes, from two or more specimens, we can choose the one and we can treat them as a lectotype for the nomenclatural type. Right? See, the next one is the neotype. Now, what is the neotype? It is the specimen or the other element selected to serve the nomenclatural type as long as the material on which the name of the taxon was based is missing. Okay, so if the material, for example, you are using the one material to name this plant, the name of the plant is based on this particular material, and if this material is missing, okay, if this material is missing on which the nomenclatural type is dependent, then the other plant material can be used to name the plant. Then in such a case we call that material as a neotype. So what is the neotype? It is the specimen or the other element. It's a specimen or the other element which is selected to serve as a nomenclatural type. We select it to serve as a nomenclatural type if the original material on which the nomenclatural type is based, if it is missing. Okay, then in such a condition that plant material we called it as a neotype. Okay, the next one, the seventh one, is called as the topotype. Now, what is the topotype? It refers to the specimen collected from the same locality from where the holotype is collected. Okay, so holotype is collected from the one locality, and after that you are going and collecting the same material from the same locality, then it is called as the topotype. Okay, so topotype is the similar type of holotype which are collected two times. So first time you have gone to the place A and you have collected the holotype, but again you have visited second time it and collected the same material, then that same material is called as the topotype. Okay, now see, The fourth one, okay, so under the typification we have considered the 7 different types, okay. But the fourth rule and regulation, fourth rule of the ICBN is the effective and the valid publication. So what is the effective way and the what is the rules regarding the valid publication? These are also given by the ICBN. Let us consider them. See, the new name of the taxon is considered valid or effective for the publication only when it is distributed in the printed form to the general public, okay. Now whenever we say publication, now what is the meaning of publication? When it should got printed, okay, digitally it should got printed and it should be available to the public. Whatever the general public is reading this botany material, this should be available to this general public in the printed form. Then and only then we consider that this particular plant material is got published, okay. Handwritten publication should not be there, and it should not be like that that this particular paper, research paper or the publication is not available for the general public. Then in such a case the publication is not valid. When the publication is valid, it should be printed and it should be available to the general public, okay. So so the research paper or the material should be in the printed form and it should be available to the general public. Then and only then it can be considered as a valid publication or the effective publication, or at least to 10 well established botanical institutions, okay. If not to the general public, at least 10 botanical institute, reputed 10 botanical institutions should have this access to this particular publication material with libraries accessible to the botanist generally, okay. So what are two criteria? The first criteria, that it should be printed and it should be available to the general public, or it should be printed or and available to at least 10 botanical institutions where the libraries are there and botanist can access this plant material. Then and only then we say that this particular plant material is got published, okay. So regarding the publication, the effective publication or the valid publication, what are the rules and regulation? The publication should be in the printed form and it should be available to the general public, or it should be available to the 10 established botanical institutions with the libraries, okay, so that botanist can assess it, right. See, the fifth rule regarding the ICBN is the author citation, okay. How to give the author citation, okay. If the author is publishing a new taxon, how he should give his own name to this particular taxon. See, the name of the taxon, unitary, binary or the tertiary, is incomplete unless the name of the author or the authors who have validly published the name, okay. So whatever the name is there for this particular plant material, without the name of the author, this particular name is insufficient, or say it is incomplete. So author citation is compulsory, okay. So whatever the author is there, he should cite his he should cite it, and after giving the citation the name is accepted, okay. Without the citation the name is not accepted, right. So the authors who have first validly published the name cited along it, okay. So for example, Mangifera indica, after the Mangifera indica there should be the L because it was first described by the Linnaeus, okay. So the author citation should be there, right. This helps in verifying the dates of the publication and impairing the precision in the biological nomenclature, okay. So what are the advantages of it? We can actually verify the dates of the publication, and whatever the imparting precision in the biological nomenclature is there, it can be imparted, okay. So the author citation, without the author citation, if the plant material is published, it cannot be considered as a valid. There should be the name of the author, that who is publishing that particular plant material, okay. Who has found this particular new plant material, who has described it, and what are the dates of the identification and description of this plant, and which author or the which authors. There may be the many authors, okay. One author, two author, three authors can be there. So all of their names can be cited, and without this citation, this particular plant material publication will remain incomplete, okay. So author citation will always be compulsory thing while publishing the new plant material. See, usually the names are cited in the abbreviated forms but never underline or type in italics. See, very important. When we talk about the binomial nomenclature in the Mangifera indica, if you take the example Mangifera indica, the first name is the generic name, second name is the species name. Here the Mangifera's M is capital while Indica's I is small, right. And the author citation yearly is given here. Now we have talked about that rule in which we have to underline the name separately if we handwritten it, and if we type it, we should make it italic, okay. But this rule is not applicable to the author's citation. You should not underline the name of the author. That is the L dot here. You should not underline it if it is handwritten, and if it is printed, you should not make it italic, okay. Which portion to make italic or underline? only the Mangifera indica. This portion you should make italic if printed. If handwritten, you should underline it separately. But this rule is not applicable to the author citation, and that is why here the Mangifera indica L dot L should not be italic or underline. Okay, so these are the strict rules. Okay, and this is very important from the UPSC mains examination point of view, because if you written down the Mangifera indica L dot and you have underlined it completely like this, the examiner at a first glance will come to know that you don't know anything about the nomenclature, and complete negative marks will be there. Okay, so you should not make such a mistake, because botanist, the typical paper evaluating system is there, and the majority of the time taxonomists were there, and if they were find such a kind of mistake in the botanical name, they will certainly make up their mind that the student is not very well aware with the botany studies. Okay, so you should not make this mistake. The author citation should not be underlined or the italics. Okay, well, rest of the name you can underline it when written, handwritten separately, and if it is printed, you can make it italic. Okay, see the choice of the name. The under the rules of the ICBN, we have the choice of the name, that which kind of name should be given. See, when the taxon rank is changed, for example, the species become genus. Okay, sometimes this happens. The species become genus. The earliest legitimate name in its new rank is the correct name. Okay, so whatever the earliest name is given to the species, that species name should be used as a genus name. Okay, so if there is a taxon, or the taxon rank is changed, okay, if the taxon rank is what's changed, then we should for example, the species, if it becomes a genus, we should use the earliest legitimate name, whatever the given to this new rank, that will be considered as a new correct name. Okay, see, when two or more taxa of the same rank are united into the one, if okay, so we have the two or the more taxa, okay, and they got united together due to their similar characteristics. For example, two or more genera were got united. Okay, two or more genera are combined together to create the one genera. Then the oldest legitimate name, whatever the oldest name is there, this oldest name of the genus should be used to give the name to this genus. Okay, and these taxa should retain as the name of the united taxon. Okay, so for example, there is a genus, there is a B genus. The A genus is having the name the B genus, and when this A genus and B genus were combined together, the name of the A should be given, because this was the earliest name which was given to this particular genus. Okay, so in this way you can choose the name, okay, while classifying the plant or while giving the new ranks, right? So this is all about this topic. Okay, thank you very much for watching this video. Please like this video, share this video with your friends who are studying the botany, and if you haven't subscribed to the Botany channel yet, please subscribe to the Botany channel for UPSC examination. Again, thank you very much for giving your time. See you in the next video lecture. Thank you.